Good morning. How you doing? I got my question computer over here, so I'm gonna look over there, and then I'm gonna get back to you. All right. First question is from Kirsten, who asks, "Do you have any information or ideas on this research?" And it's a link, and the link goes to an article about a guy called Magnus. I'm sure I'm gonna mispronounce it. It's Hook, but there's umlauts over the O's, and it's Swedish. I'm gonna guess Hook, something like that, but I don't know. I apologize, Mr. Magnus. Anyway, what the article is talking about is the start of a clinical trial that's related to the idea is that this could be something that would work for COVID 19. And again, we are not directly attacking the virus, but this is maybe one step closer to a direct attack. So the idea is that you can give this thing, and I'll go into what the thing is in a second, and it should change the way that antigen presenting cells, which is cells like neutrophils, macrophages, dendritic cells, they're cells of the immune system whose job it is to go and hunt down something that doesn't look right and then chop it up and then display all the little parts of it, like a hunting lodge wall, all the little heads of the things that they've found. And they do this so that other cells from the immune system can learn to recognize them and can make a response to them. And so they'll do this on command. Rather, they'll just go around eating stuff, and then when they get a certain signal, then on command, they will just start to burp up a little bit of whatever's the last thing that they ate. And so the point of these signals is that these are kind of generic markers of danger or of something else going wrong. It's like the second key you turn at the same time as the first key to launch a nuclear missile. You got to have two signals that something weird's going on, and then that's usually good enough, and it's okay. Let's make a response. So this thing is called. This was a mixture of two different things. One is called PAM2CSK4. Yeah, which is a code that tells you exactly what it is. So this is a lipopeptide, which is a weird little thing that's part lipid, like the things your membranes are made out of on the cell, and part peptide, which is like a little baby short protein. And this is both. It's got 6 amino acids, which are C, S, and then 4Ks, so that's the CSK4 part at the end. And on the other end of it, it's got 2 myristic acids, which are the fatty acids, and so that's the M2, I think, at the beginning. I didn't look into it, but that would totally make sense. And there are actually a whole bunch of these things that have been investigated over the years. Bacteria make them, and bacteria are very weird. They make the weirdest little proteins you'll ever see. And they've got special ways to make some of these real oddballs, because you can't just make them in a normal way with messenger RNA going to the ribosome, just like you learned in school. So that's one of the components, and that one's supposed to stimulate these two receptors called toll-like receptor 2 and 6, which are things that are expressed, for example, on macrophages and on various other cells in the immune system. TLR2 and 6 would potentially activate these and get them to start basically coughing, like when you tell a dog, drop it, leave it. Yeah, like that. Just like, what'd you get, boy? Yeah, let's see. Come on, show me. The other thing they're putting in is a thing called CpG. So it's a little tiny bit of DNA that's the nucleotide C and then the nucleotide G. And they're right next to each other, and they're hooked up via a phosphate, and that little bit is also enough to set off a different toll-like receptor called toll-like receptor 9. I think I've got the receptors and numbers right on this, but same deal. These are different ways to activate potentially different kinds of cells. So the first paper I saw on these is, or at least on their action, is from the mid-2000s, like '05. Their discovery probably goes back before that. Yeah, it looks as though this Mr. Magnus's lab might have got involved around 2011. And yeah, so now they're pushing these out there. I could see ways that it could work. So if it worked exactly like we thought, then this makes some sense. And you're giving macrophages, which are kind of going off and causing disease, you're giving them a job to do. And you're trying to, It's like one of those I don't know 80s scared straight programs or whatever where they used to lock kids in jail. It's trying to give these macrophages something to do so that they don't make trouble. I think maybe it's more like a youth center. Let's call it that. That's nicer. Yeah. Now, of course, these are molecules that bind to receptors that are on macrophages. And so the risk is that this is how we figure out that they maybe stimulate macrophages and maybe make things worse. People have deliberately given these, at least the first one of these compounds, the lipopeptide, in order to try to convert macrophages to a more anti-tumor state, from what I'm seeing in like the couple of papers that I've skimmed this morning. So the question is that gonna is that gonna be good or bad in COVID? And I think we do not know that yet. Right now, they're just doing safety testing. It's potentially a powerful way to do something, and the something could be good. Yeah, that's about all you can say about this now, but it's a thing that works in a way that has a plausible means of doing something in COVID. And at this point, yeah, that's better than just, throwing compounds at the disease until we find one. At least you've got a path that might work. So that's as far as I can tell about this particular research. Looks kind of neat. Yeah, interested to read more about it when it comes out in the future. So there we go. It's a bit long, but there's a lot of weird stuff in there. So thanks very much. This is Ask Dr. Ben.